really an honor to be here. Thanks for the invitation. It's really a very interesting crowd as well, so I'm very pleased to give this talk. I slightly modified the title a bit from computational genomic approaches to omics approaches, because after some discussions yesterday I decided also to include some computational metabolomics, which we've started doing in my group within the last year as well. But basically what I want to talk about is how can we leverage all these large scale genomic and metabolomic data sets to accelerate natural product discovery and get a better handle on all this metabolic diversity to really target antibiotic discovery. Because it was already clear from David's talk and Andri's talk that there is lots of diversity out there, but there are still quite a number of challenges on how to really utilize and exploit that diversity. So for us, a key handle of understanding natural product diversity and chemical structure diversity is by looking at the genomes of their producers. And the key units in this are what we call biosynthetic gene clusters. And it's really nice to see and very useful to see that often what you see in the chemistry of different natural products is reflected in the composition, the gene composition of these gene clusters, which encode all the enzymes that are necessary to build up and construct these molecules. So for example, if you look at some of the molecules that are here on the screen, for example, ribadirid has this coumarin group, which it shares with simocyclinon, which was also shown by Andri in his presentation. And it's nice to see that actually the chemistry is shared. When you compare the gene clusters, there's actually also groups of genes shared that are actually involved in building and attaching that chemical moiety. Similarly, this nitrous sugar is shared between ribadirin and everydinomycin, and you'll see that the corresponding genes are shared between their gene clusters. And for example, this orsolinic acid is shared between polyketomycin and everydinomycin, and the corresponding brown genes here are homologous between their gene clusters. So by looking at the gene clusters, we can already get an idea of what kind of diversity is out there in terms of natural product chemistry. The first step in this process is, of course, identification of these gene clusters. And as Alexander mentioned, during my PhD, I, together with Kai Blin and Telman Weber, who were at that time in Tübingen in Germany and are currently in Copenhagen in Denmark, we developed Anti-Smash, which is a web server that automates the identification and computational analysis of these biosynthetic gene clusters. And together with them, we keep on coordinating the development of this tool, which is also publicly available on a web server, which has been used around 350,000 times now. So lots of people are using this. It uses a lot of computational resources. It basically automates the identification of these biosynthetic gene clusters. So here's an example output of Streptomyces silicolor, the model actinomycete. You see here 25 gene clusters being identified by Anti-Smash. Cluster 10 is currently selected. You'll see the genes here that in this case encode an NRPS, non-ribosomal peptide synthetase biosynthetic pathway. And for the actual NRPS enzymes, you'll also see the modular domain structure of those enzymes, with which, based on the subset specificity prediction of certain domains within those enzymes, we can already predict kind of a core structure of the molecule. So, of course, it's nice to be able to automatically identify all these gene clusters, but of course the second key step is also to be able to kind of dereplicate these gene clusters at the genetic level to see, okay, if I put a genome through Anti-Smash, the gene clusters that I find there, are they similar to any gene clusters for which we know the function? And this used to be actually a very big challenge, because the information on all those gene clusters was scattered through the literature of a few decades. So that's why during my postdoctoral fellowship in Bremen at the MPI, I coordinated a large community effort to basically pull all that information together with more than 80 research groups worldwide. We did this, and we basically standardized the way in which these gene clusters can be annotated, and according to that new data standard for annotation of biosynthetic gene clusters, we together also annotated more than 1,200 biosynthetic gene clusters, and since then the MI-BIG, minimum information on a biosynthetic gene cluster repository, has been updated to now contain almost 2,000 biosynthetic gene clusters that have been linked to structures. So this, of course, is coupled back to anti-SMASH, so that whenever you now put a genome through anti-SMASH and you identify a gene cluster, it is compared with all the biosynthetic gene clusters in this MI-BIG repository, for which we know the chemical products, and you get a ranked list of the most similar known gene clusters to which your gene cluster of interest, the gene cluster that you detect, is similar to. So this is a way in which you can quickly see whether, for example, a gene cluster of interest is either identical or more or less identical to a known gene cluster, in which case it's not very interesting, or it is slightly different, in which case it could be interesting because it could lead to the production of a novel derivative, or perhaps it doesn't look like anything we know, in which case it could still produce a known compound like Andrie's shown, but at least at the genetic level we do not know any biosynthetic gene cluster or pathway that is similar to the gene cluster that you then find. So basically this pipeline automates a large part of the computational genome analysis of these biosynthetic gene clusters, but there is one big problem here, because the field and the development of omics data has not stood still in the past years. When I developed anti-SMASH, the way people were doing science with genomes was sequencing one genome, and then I knew at that time there was one group in the US that sequenced like a dazzling number of 20 Streptomyces genomes, and it was just amazing. And currently that's just peanuts, because people are looking at much bigger data sets. So for example, with Alexander and Jan Song from Syntef, we yesterday had a project meeting with their project in which we're looking at, well, here it says 1,000, but now with some additions it's now even more than 1,200 actinobacterial genomes. We're talking about more than 25,000 biosynthetic gene clusters. So the way to study this is, well, we need something more than just anti-SMASH, because looking through 1,200 outputs of separate outputs of individual genomes and trying to make sense of that is just undoable. It would take months of boring work, and you would probably not be able to find the really interesting bits. We need new ways to be able to look at that data. And it's not only the large numbers of genomes that are a problem for this traditional approach. Also, metagenomes are delivering very large numbers of biosynthetic gene clusters. So I think David already gave a nice example of a large metagenomic data set of 256 gigabytes. There was another recent study by the Bork lab where they sequenced also like 200 soils from all over the world, enormous masses of sequence information that could potentially be leveraged to look at new biosynthetic diversity. So there's lots of those data sets coming out there. One other bottleneck you have with metagenomics, especially with the shotgun metagenomics, is that it's very difficult to assemble complete biosynthetic gene clusters from those data. Basically because of the complexity of these microbial communities, but also because many of the biosynthetic gene clusters are very repetitive. For example, polyketide synthases and non-ribosomal peptide synthetases have kind of repetitive domain structures that make it very difficult to assemble them. So we've recently worked with a group of Pavel Pevsner in He has actually a lab both at UCSD in San Diego, also in St. Petersburg, to come up with a new assembly algorithm that is specifically focused on being able to complete these kinds of biosynthetic gene clusters. So actually what you get when you normally assemble a complex metagenomic assembly are small contigs. These contigs are taken from an assembly graph in which, well, I will spare you the computational details, but based on K-mers, kind of you get stretches of DNA and it can be linked to each other. And for example, this stretch from here to here would be a large contig. And for example, this gene cluster here, which encodes the biosynthesis of a lipopeptide, in the assembly graph is actually split into 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 separate contigs. And normally if you don't look at this graph, which nobody really does unless you're really good at this kind of stuff, you would just look at these small separate contigs and you would have no idea that they belong to the same gene cluster. But now this biosynthetic spades algorithm, which was developed by Dmitry Meleshko from Pavel's group together with PhD student in my lab, Vitolia Takana, it basically looks at these graphs and reconstructs the most probable paths here from basically the start of the gene cluster to the end of the gene cluster. And in this way, it's able to kind of reconstruct the most likely actual sequence of the full biosynthetic gene cluster. And based on reference genome information, if sometimes you have multiple possible paths through the graph, it can basically pinpoint the most likely order of those contigs and the pieces in between. So you get really complete gene cluster information. And we've tried this on a number of metagenomes, and this really allows you to get large numbers of pretty complete biosynthetic gene clusters from metagenomes. So what this all means is basically using these large-scale strain collection, genome collections, large metagenomes, we get access to thousands of biosynthetic gene clusters, and this is enormous diversity to wade through. And we need targeted approaches to be able to get a bird's eye perspective of that diversity and to be able to explore that effectively. So one technique that I developed together with Peter Chiramancic at Michael Fishback's lab when I was visiting Michael's lab in San Francisco during my PhD is basically a technique using sequence similarity networks. So in this is a network of all the biosynthetic gene clusters in around 1,000 genomes. And here each node represents one biosynthetic gene cluster. And here the colors of the nodes indicate what biosynthetic class of gene cluster each node represents. So the nodes are connected based on a similarity metric. So when two gene clusters are more similar than a certain cutoff, then they would be connected. And the similarity metric here includes several items such as domain content, protein domain content of the gene clusters, their order, and their sequence identity optionally. And by doing this, you can also use the known gene clusters, currently the ones that are in MI-big, put them into the network, and you would actually be able to see that, for example, this is a region where we already know most of the biosynthetic gene clusters, where, for example, here is another region of the biosynthetic space where we haven't really characterized any biosynthetic gene clusters experimentally. So this allows you to identify and target kind of unknown spaces of biosynthetic diversity. So another thing that you're able to do with this is using the same kind of distance metrics, and there was actually several labs that have in parallel constructed similar distance metrics. You're able to group large numbers of biosynthetic gene clusters in what we call gene cluster families. So now each gene cluster family would basically contain all the biosynthetic gene clusters that make either the same or highly similar natural products. And this also allows you to get a better grip of the diversity and also see, for example, which strains have similar gene clusters of a particular kind. So In the past 2 years or so, or 3 years, we've kind of completely automated this approach into a fully comprehensive workflow that interacts smoothly with our anti-smash biosynthetic gene cluster identification tool and the MIBig reference database. And we launched 2 new tools called Bigscape and Corazon. Corazon was built by Nelisea Moica from the group of Paco Barana Gomez in Mexico. And so what happens here is first you have a bunch of genomes, you run them through anti-smash, you get all your gene clusters, you add reference gene clusters of known function from MIBig, then you run them through Bigscape, and Bigscape automatically reconstructs sequence similarity networks of all your gene clusters, including the reference clusters, and groups them into families. In some cases, they will be families without reference gene clusters, so families of unknown function, and in some cases, they will be families with reference gene clusters that do have gene clusters of unknown function. And then subsequently, what Corazon can do for you is take, for example, a gene cluster family of interest and reconstruct a multi-locus phylogeny of all the members of all the gene clusters within that family, so that you can see how all those gene clusters are actually evolutionarily related to each other, to basically look at their diversity at a higher resolution. So to be able to make this really smooth and to be able to make this doable, we also needed to make a number of computational improvements, because in the original studies that launched these sequence similarity network and gene cluster family approaches, if you would take 1,000 genomes, you would basically need a supercomputer to crunch these data for 2 months or something, and then you would get your data back. But we wanted this to become available for everyone, and we wanted to make it possible to run it on your own laptop within a few hours at most. And I'm very glad that in the end we're able to do this through a few tricks that were already there previously and a few new tricks. One key trick is that basically from a sequence of nucleotides we compress gene clusters to a string of protein domains, which are kind of the functional, which represent the functional content of each gene cluster. And then we have distance metrics that basically compare these gene clusters, for example, first by looking at the protein domain content in one gene cluster and another gene cluster and looking for the shared portion. We can do the same to look at gene order by looking for pairs of adjacent domains and looking how many adjacent pairs are being shared. But the most tricky part was using sequence identity, because for the previous approaches, the supercomputers were especially needed to be able to align like thousands of protein domains to each other in big all versus all multiple sequence alignments, and this just took ages. So the problem here is that this isn't a bit of computer science. This is an all versus all problem, and this scales quadratically. So if you add more gene clusters, you would get quadratically more compute time, because the number of comparisons in a two-dimensional matrix would scale exponentially, well, with at least an exponent of 2. So we've basically changed this by going from an all versus all comparison to comparing all versus a profile. So for each of those PFAM domains, there's basically a model, a sequence model that basically can function as a reference for the alignment. And then all what you then need to do is take this sequence model, and then you align one sequence to it, another sequence, another sequence, another sequence, etc. So because you don't align the sequences against each other, but only against this model, it's called the profile hidden Markov model, this makes it from an all versus all problem into an all versus this single model problem. And this key trick sped up especially the large searches from months on supercomputers to 2 hours on a laptop computer. So actually this weekend I ran an analysis on the 1200 genomes from Syntev, and it took, on my laptop computer over here, it took about 3 to 4 hours to look at all the like 25,000 biosynthetic gene clusters. So that's pretty quick, I would say. Finally, one final trick that we implemented has to do with comparing gene clusters, and especially in metagenomes and in draft genomes, often the clusters are fragmented, so they still end at a contig edge, even after the tricks that we can do with biosynthetic spades. So the problem there is if you compare fragments to complete gene clusters, it's difficult how to really make a fair comparison. You cannot really compare globally, like the whole gene cluster versus the fragment, and only look at the shared part. But you also do not want to basically look, only look at the, so if you compare locally, you would only look at the shared part. If you compare globally, you would penalize the fragmented gene cluster for not having all the other domains that are actually not part of it due to the fragmentation. So the solution for this is what we call glocal alignment, in which we first look for the best local match very quickly using a fast algorithm called the longest common substring search. And then we extend the alignment to two sides, to the nearest gene cluster border, which could be a contig border. And then we look at the corresponding pieces in both gene clusters so that you really compare the part that should correspond, but not only the local best matching part. And this is a nice way that kind of hovers in between the global and local alignments and really works well to also align the MI-big gene clusters to the anti-smash clusters. So we validated this, of course, on a large scale by looking at 248 natural products from MI-big clusters that we could group into, based on literature, into 93 curated groupings. And we saw that Bigscape was able to accurately recapitulate those groupings. And this also allows you to basically look at the data and see, for example, if you have gene clusters of interest, whether they will collate, for example, with gene clusters of known product classes. And in this way, these four gene clusters here would be new, possibly new glycopeptides. At least they warrant a further look if you're looking for new glycopeptides. So it's of course important to also make the data analysis then accessible. And for that we, and particularly Satria in my group, build a user interface to be able to explore the output. So you get an overview of here gene cluster families versus genomes. And then you can go, for example, to the network of a particular product class, for example, the type 1 polyketide synthases. And here you get a visualization of the network. You can select certain gene clusters of interest or hover over them to see their names, or look, for example, for a gene cluster of interest from MI-big, a reference cluster, which will then highlight. You'll see the corresponding gene cluster family around it. And then you can go to the family, and here you get the Khorasan phylogeny of all those gene clusters to see how they are related to each other. And when you hover over them, you see the protein domains, etc. And in this way, you can very quickly explore all of these networks. So Bigscape has already been quickly adopted by the scientific community. There's lots of labs that have already started using it even before publication, so we're happy with that. This is a nice example of the groups of Jörn Piel and Julia Vohal that used it on the plant microbiome dataset. And so for Khorasan, which makes these phylogenies, there's one other thing that is really cool that you can do with this. You can also take any gene of interest from your gene cluster of interest, then basically look for all the different contexts in which you find this gene together with at least one other gene in the gene cluster, and identify, for example, different gene cluster families that have this gene together with at least one other gene in the cluster, to be able to look at how that particular enzyme family evolved in what kind of gene cluster content. So to be able to look at this in And in a case study to see how we could use these tools, we looked at a natural product family called the ditoxins and ramosamides, together with a group of Neil Kelleher, and in particular the postdoc there, Michael Maloney, who worked at this. And he had seen from mass spec data, and this is a network that he could generate from the mass spec data, that this is a very diverse family of natural products. So there's a lot of molecular diversity, dozens of different variants across lots of different strains. And we were able to see also using Bigscape that there were actually a number of different clades in our network that correspond to different subfamilies for ramosamide and ditoxin production. And when using Corazon, we made a large tree of the ditoxin ramosamide superfamily, gene cluster superfamily. We were able to see that different kind of branches of this tree, different subfamilies, also had slightly variations in enzyme content. For example, this Amycalotopsis clade has certain P450 hydroxylases that the others do not have, and the clade here also has certain tailoring enzymes that these two known clades do not have. And based on mass spec and NMR analysis, we were able to see that indeed all of these clades lead to the production of different variants of those molecules. So what's also really useful is that you can use these gene cluster families and, for example, also correlate them, correlate the presence of members of a gene cluster family with the observation of specific peaks in your mass spectrometry data. And this is what the groups of Neil Kelleher and Bill Metcalf have done for a while now. And also we've shown together with them that when we use Bigscape, this also works well for many natural products. So we get a strong correlation score between observing a specific mass peak for those molecules and observing their gene cluster. So basically the idea is if you very often find a certain mass peak of interest in the same strains where you also see a particular gene cluster, in many cases that's not by chance, but it's because the cluster is responsible for making that molecule. And this you can use to prioritize. But we can do more than that. Using mass spec data, you can also create networks similar to what we can do with gene clusters. You can create also molecular networks, and this is based on the fragmentation pattern of these molecules. So what you can generate are tandem MS spectra, in which you kind of get an idea of the fragments that arise when you shoot on those molecules, and you fragment them using ionization. And then based on the similarities between the spectra for different molecules, you can group the molecules also in a network in which the differences in a network, the nodes would usually also correspond to distinct mass shifts or mass differences between different molecular variants. And now, of course, the big challenge is to be able to kind of compare the molecular networks with the gene cluster networks. So to link families of molecules to families of gene clusters at a large scale. So there's two possible ways to do that. So first of all, of course, we have the sequence data. We predict the gene clusters. We cluster them into gene cluster families. With the MS data, we have MS data. We use spectral clustering and generate molecular families. And then there's two ways to map those to each other. The first is correlation, which is similar to what I've shown before, but then looking for correlation not between gene cluster families and specific mass peaks, but between gene cluster families and molecule families. And the second is feature-based mapping, in which you predict from the gene clusters certain chemical features that you can predict are going to be present in the products, and you try to link that with chemical features that you can predict based on, for example, fragmentation patterns and mass shift in the MS data. So this we call feature-based mapping. We're currently developing this technology. A key step in this technology is, of course, that you need to really annotate these networks to identify these chemical features. And for example, Justin in my group has been working with Kyo-Min Kang and Madeleine Ernst from Peter Dorrestein's lab on a new method that takes molecular networks and uses motifs, specific motifs that are linked to specific substructures in those molecules. For example, this would be, or the green one here would be a motif of two peaks that are linked to vanillic acid. So if your molecule fragments and you get those peaks, it's very likely that you have a vanillic acid in your molecule. And using those motifs, you can annotate the networks and identify where certain substructures are found in which molecules. And this has now been also generalized in a pipeline in which you have MS-MS spectra. You do this MS-MS molecular networking, and then you use both the motifs and also reference spectra. So actually the spectra for known molecules that have been annotated in this GNPS database from UCSD. And these reference data you can also use to basically get a chemical classification of, and that's indicated here by the colors in the molecular network, chemical classification of structures using an algorithm called classifier. And when you combine the motif analysis with these chemical class level annotation, you really get highly annotated molecular networks, also with a lot of molecular features. At the same time, we can do something similar for gene clusters. For example, together with Jörn Piel's lab, we've generated an algorithm that's able to predict specific, basically the structures for trans-AT polyketides and look at their diversity based on how the domains involved in putting together those molecules, how they clade in their phylogenies. And we also develop machine learning approaches that are able to predict the substrates for non-ribosomal peptide synthetases, the amino acids that are being incorporated in these NRPSs. And we've already shown together with the lab of Peter Dorrestein that you can use this information to link lots of basically molecules from large-scale metabolomic data to gene cluster families. So Peter Dorrestein's group has recently done large-scale metabolomic profiling of a few hundred Pseudomonas strains, and those contain a number of lipopeptides that are known to be also antimicrobial in many cases, or antifungal. And there were in the molecular network a number of new masses that were clear from the molecular network that they were different from the known molecules. So here the blue ones and the orange ones were known families, and the purple ones and these red ones were new, so they had not been annotated before. And using an algorithm that matches the mass shifts for the amino acids in the mass spec data with the predicted amino acids in the gene clusters, we were able to link those molecules semi-automatically to gene clusters. So the next step, after being able to predict and link monomers like amino acids between molecular networks and gene cluster networks, is also going further by predicting at large scale also tailoring reactions from gene clusters. And the biggest difficulty here is that we also need to predict where those tailoring reactions will actually do their thing on the natural product structure. The good thing is that there seems to be quite some phylogenetic signal in this regioselectivity. So for example, if we have here the WS9326 biosynthetic gene cluster, and we have an O-methyltransferase that here methylates this tyrosine here in the structure, and we look at other NRPS gene clusters where we see a closely related O-methyltransferase. Here in the pheglymycin, the pheglymycin gene cluster, this is the marformycin gene cluster. We see a similar methyltransferase that on a completely different molecule also methylates the hydroxy group of a tyrosine. So we can actually use this and other information to also try to predict the regioselectivity for those kinds of enzymes. At the same time, we often see that gene clusters contain, like I showed at the start, also different subclusters that encode the biosynthesis of specific moieties that are incorporated in these natural product structures. And we see that those can also evolve into different variants that are linked to different chemical variants of those of those chemical substructures. So for example, we see here different variants of those subclusters that, for example, have or do not have these methyltransferases. And for example, having a methyltransferase or not would make the difference between this 3HA or the 4MHA. And using the variation between subclusters and substructures, we're now trying to automatically predict those chemical features. And ideally, when we then predict the structural features from the gene cluster, so we get a list of structural features that are predicted, we can link those, we can match them to observed structural features. And in many cases, we can see, even though our predictions are not perfect, that there is a good match that is really beyond chance. And then we can start learning from the data, so saying that this would be our prediction based on the gene cluster, this would be what we kind of observe in the metabolomic data. We can then go back and retrain the algorithm by saying, okay, our predicted structural features were not perfect, so the phenylalanine was actually another aromatic amino acid, it was actually a tyrosine. This one we had no clue what it was this exotic amino acid quinine. So in this way we would be able to iteratively learn from the data and improve those predictions to really combine genomic and metabolomic data and effectively really exploit those large kinds of data sets. Okay, with that I want to end and thank the members of my research group, in particular Jorge, who worked on Bigscape, Barbara, Satria, Justin, and these master students over here who worked on the work that I presented today. And I want to thank you for your attention.